The next important topic is regulation of gene expression. Gene expression I have told already. So it is the protein synthesis. It refers to protein synthesis through two major events, transcription and translation. This gene expression is regulated. Why it has to be regulated is there are 20 different amino acids which constitute different proteins. Synthesis of all amino acids requires energy, but all the amino acids which constitute proteins are not needed at the same time. There is need to control the synthesis of those amino acids which are not required. Therefore, a control system is operative, which is known as gene regulation. So, whichever amino acids are not required has to be controlled. For that, the gene has to be regulated. So, that is why we have regulation of gene expression. This is extensively studied in prokaryotes, especially in E. coli, which is found in the guts of human beings. This regulation of gene expression can be controlled at either in the transcriptional or post-transcriptional level or even in translational level. Transcriptional level is during the process of transcription, that is when the information in the DNA is copied to the mRNA, or after transcription process, that is post-transcriptional level, or translational level, that is after transcription, when the mRNA leaves the nucleus and the formation of protein occurs. So either it can be in the transcription or post-transcriptional or translational level, this regulation of gene expression can be done. We are going to discuss it in the transcriptional level. For this, there is a small extracellular or intracellular metabolites which can initiate or inhibit gene expression. Metabolites means there are primary metabolites and secondary metabolites. Primary metabolites includes lipids, proteins, carbohydrates, vitamins, all these things. So, any of these can act as a metabolite which can either initiate or inhibit gene expression. In this, we are going to learn a term called operon. So, operon means the cluster of gene with related functions. So, you can say a group of gene which are more or less having the related functions, they are called operon. So, these operon, they transcribe single mRNA molecules. There are nearly 260 genes grouped into 75 different operons in E. coli. So, how many genes are there? Nearly 260 genes, and these 260 genes are grouped into 75 groups, that is operons, and this is in E. coli. We will see the structure of the operon now. So, this is the diagrammatic structure of operon. So, it consists of structural genes, an operator gene, promoter sequence and regulatory gene. So, a DNA sequence, this is a DNA sequence with all these factors, then it is called as operon. So, what are those that present in the operon is structural gene, operator, promoter and regulatory gene. These are also a sequence of DNA, but they have some related functions. So, they all together they are called as operon. So, it consists of one or more structural genes. So, what are structural genes? Structural genes, it code for the synthesis of specific polypeptides. Not only polypeptides, for tRNA, rRNA, whichever are required by the cell. So, these all are coded by the structural genes. And next one is an adjacent operator. So, next to the structural gene, adjacent is the operator gene. This operator gene contains the code which is necessary to begin the process of transcribing the DNA message of one or more structural genes into mRNA. So, all those information that is required for the process of transcribing the DNA message, the information present in the DNA into mRNA, that is all only with the help of this operator gene. So, that code is present in the operator gene. Next one is regulator. So, this regulator gene, these all are genes. This regulator gene, it is involved in controlling the expression of one or more other genes. So, these all together they are called as operon. So, promoter is the gene sequence where the RNA polymerase binds and the transcription begins. So, these are all together called as operon. So, this is the structure of operon. Structural genes are linked to an operator gene in a functional unit called an operon. Ultimately, the activity of the operon is controlled by a regulator gene which produces a small protein molecule called a repressor. this regulator gene, it produces a protein molecule, and that protein molecule is called repressor. You should be aware of all these terms, only then when I explain the video it will be clear for you. So here the activity of the operon is controlled by regulatory gene. So if the operon has to work, that is if it has to be on or off, is only controlled by the regulator gene. So this regulator gene, it produces a protein molecule called as repressor. The repressor binds to the operator gene and prevents it from initiating the synthesis of the protein. So I told you it produces the protein molecule, and if this repressor binds to the operator gene, the entire process of protein synthesis stops. So and prevents it from initiating the synthesis of protein. So if the repressor is binding to the operator gene, the particular function of protein synthesis stops. The presence or absence of certain repressor molecule determines whether the operon is off or on. So the repressor binds to the operator gene and prevents it from initiating the synthesis of the protein. The presence or absence of certain repressor molecule determines whether the operon is off or on. So this is what repressor molecule, this is what will determine whether the operon has to be functional or non-functional. So I told you there are many operons. In this we are going to see about the Lac Operon, which is seen in E. coli. The Lac Operon is also called as lactose operon. So this involves the metabolism of lactose in E. coli. And the Lac Operon is an operon or group of genes with a single promoter transcribed as a single mRNA. The genes in the operon encode proteins that allow the bacteria to use lactose as an energy source. So in Lac Operon, the energy source is lactose. So based on the energy source, the operons change, become operative or non-operative. So here when the bacteria use lactose as energy source, the Lac Operon becomes active. So we will see a video for this Lac Operon. In this video we are going to understand about a very important topic, the gene regulation and the Lac Operon. So let's start with gene regulation. Gene regulation is the most simplest term, is the ability of the cell to control or regulate what proteins it makes from its DNA. In our body, we have numerous types of tissues and cells. We have muscle cells known as myocytes, we have cells in our intestine known as enterocytes, and the cells in our skin known as keratinocytes. All these cells are different in shape, size and functions as well. The myocytes' main job is muscle contraction, enterocytes produce enzymes for food digestion, and keratinocytes form a barrier known as skin. All these cells contain the same DNA in them. However, they are different in sizes, structure and function due to the different proteins they produce. As most of you may know that our DNA is made up of thousands of nucleotide bases which are paired, and these are the basic units of DNA. The human DNA contains over 3.1 billion base pairs. These base pairs are arranged in groups known as genes. Now if you take out the DNA from the nucleus of a muscle cell, a skin cell and a cell from our intestine, they would contain the same 3.1 billion base pairs of nucleotides arranged in the same sequence. But despite having the same DNA in them, they are completely different. And this is because of the different proteins they produce. This is all possible due to the ability of the cell to control or regulate what proteins it makes from its DNA. In a broad sense, the genes in our DNA can be divided into two main groups: structural genes, which codes for proteins, and regulatory genes, which regulate the proteins that are produced by the structural genes. We have about 20,400 structural genes in our DNA that codes for various proteins. But we will be supposed to know that that makes up only 2% of the amount of DNA. The vast 98% of our DNA does not code for any proteins, and they are mostly regulatory. And this non-coding regulatory DNA are responsible for gene regulation. To this process of gene regulation, a cell can also increase or decrease the production of a specific protein. For example, the B cells in our body produce antibodies that fight off infection. The antibodies are also proteins and are produced at a low rate of production in the absence of an infection. But in the case of infection, the production of the antibody can be increased many times over, all through the upregulation of genes that are produced by these antibodies. Another common example includes the beta cells in the pancreas that produce the hormone insulin. Now these cells do not produce insulin at a constant rate, but rather increase or decrease according to depending on the amount of glucose level that is present in our bloodstream. This is possible because of the Bt cells of pancreas have a special receptors in them that enables them to sense the glucose in the blood. But when the blood glucose rises, it is noted by the receptors and sends the information to the DNA. The regulatory genes located behind the insulin gene note this and increase the amount of insulin that is produced by the structural genes. This is one simple example for gene regulation. Gene regulation works in thousands of ways, but we are going to see the basic model of gene regulation that operates in bacteria that is known as E. coli, Lac Operon.